Okay, welcome to chemistry classes. In this video, we have to discuss here separation of mixture, filtration, distillation, chromatography, and sublimation. This type of different type of separation of technique we have to discuss here. Okay, so we know there are different type of mixture, homogeneous mixture and heterogeneous mixture. We discussed in previous video, we discussed heterogeneous mixture and homogeneous mixture. Heterogeneous mixture means different components of mixture do not mix homogeneously. That is heterogeneous mixture. Example for heterogeneous mixture: sand in water, chalk powder in water. Heterogeneous mixture. Homogeneous mixture: different components of mixture mix homogeneously. Example: salt in water, sugar in water. These are example for homogeneous mixture. Heterogeneous mixture we can separate very easily. For example filtration, sieving, this type of technique we can use. Okay, heterogeneous mixture we can separate by simple hand picking method, sieving, filtration. Simple hand picking method mean we have one heterogeneous mixture, we can simply separate with hand. Some type of mixture we can separate such like this. This type of method is called simple hand picking method. Clear? Okay, after this we have to discuss here filtration. I told you heterogeneous mixture, example sand in water, we can separate by filtration. For filtration we will use filter funnel. We will use this type of one funnel. This is called filter funnel. On the top of filter funnel we will keep filter paper. Okay, and we will put the mixture from the top of the filter funnel. All solid will stay over here, only liquid will come down. The solid which stay on the top, it is called residue. For example, when you put sand in water, when you put sand with water on the filter funnel, sand will stay over here, only water will come down. The sand which stay on here, the sand or residue, the residue which stay over here, it is called the solid which stay over here, it is called residue. Water, the liquid state which came down, it is called filtrate. This method is called filtration. So the special apparatus used for filtration, filter funnel. Okay, so remember here filter funnel, residue and filtrate. Okay, and this is one example for heterogeneous mixture. Okay, next one we have to discuss here separation of colored components from blue or black ink. We have blue or black ink. From this ink we have to separate different components of mixture. Okay, so here we have to discuss which are the different procedure to separate different components of colored components from ink. So for this purpose we have to take a beaker with water and cover the beaker with a watch glass and put few drops of ink on the watch glass. So see here in this diagram, here we have to take a beaker with water and cover the beaker with a watch glass and put few drops of ink on the watch glass, then heat water. Okay, do not heat ink directly. Okay, do not heat ink directly. We have to keep watch glass over here and heat water in the beaker. Okay, because of this heat ink from watch glass get hot and it start to evaporate. Okay, after finished evaporation from the ink, we will stop heating and we will get residue from here. We will get residue from the watch glass. So here ink is a mixture. Ink is a mixture. Mixture means impure substance. We discussed in previous video. Mixture, it is impure substance. Compounds, the substance which contain only one type of substance, only one type of elements or only one type of compounds. These are pure substance, but mixtures are impure substance. So see here, ink, it contains different colored components, also something evaporated over here. So here it is mixture. Okay, so this mixture we can separate by this method. Take water in a beaker, cover a watch glass on the beaker and put few drops of ink on the watch glass and heat water directly. Do not heat ink directly. Okay, and here ink from watch glass will be evaporate. After evaporation finished, we will stop heating. Okay, then we will obtain residue from the watch glass. Okay, so this is the method to obtain colored components of ink. Okay, colored components, or you can say different dyes from the ink. Okay, we have to discuss here separation of cream from milk. How to separate cream from milk? Okay, to separate cream from milk, we will use centrifugation. Centrifugation means the mixture rotate very fastly. Okay, the mixture rotate rapidly in a particular machine called centrifugation machine. To separate cream from milk, we will use this method, centrifugation method. Centrifugation means the mixture rotate very rapidly, so we can separate cream from milk. Okay, this is the apparatus used to separate cream from milk, or this is the apparatus used for centrifugation. Okay, and here mixture rotate very rapidly in this type of machine. Okay, and here application for centrifugation. Here these are the different application in centrifugation to test blood and urine. Okay, diagnostic laboratory using centrifugation to test blood and urine sample. Okay, and another one to separate butter from cream. To separate butter from cream, also we will use centrifugation. Also another one, this method also used in washing machine. Washing machine to squeeze out water from wet cloth. Okay, to dry cloth in washing machine, we will use this method. Cloth will spin rapidly. Okay, so it helps to drain cloths, to squeeze out water from wet cloth. Okay, so these are the different applications of centrifugation. Okay, so cream from milk, butter from cream, this type of separation we will use centrifugation. Centrifugation means the mixture rotate very fastly and helping to separate cream and milk, or solid and liquid. We can separate this method. Okay, one we have to discuss here, separation of immiscible mixture. Immiscible mixture or immiscible liquids mean two liquids do not mix each other. Two liquids do not mix each other. Such type of liquids are called immiscible liquids. This mixture can be separated with help of separating funnel. Separating funnel. You can see here separating funnel. Okay. So this type of funnel we can use to separate immiscible liquid. Immiscible liquid example, oil in water, kerosene in water, they do not mix each other. These are called immiscible liquid. Okay, so take immiscible liquids in separating funnel and shake well, then keep it one place. Okay, let it stand undisturbed, means keep this in one place and do not disturb, do not shake it. Okay, then it forms two separate layers. You can see in this diagram, it forms two separate layers. After that, you have to open the stop cock. Okay, then pour out the lower layer. take out the lower layer of the liquid. This is the method to separate immiscible liquid. To separate immiscible liquid, we will use separating funnel. Okay, separating funnel. And application for this process. This process helps to separate oil and water. This process helps to separate oil and water. Also, this process applicable in extraction of iron. In the extraction of iron, also we will use this type of separation. Okay, separation of immiscible liquid. Okay, extraction of iron, and another one, separation of oil and water. Extraction of iron you will study in higher classes. Okay, during extraction of iron it form molten iron, also it form molten slag. Slag mean impurities in the iron. So there will be two separate layers. These two separate layers can be separate, okay, with help of separating funnel, because they are immiscible liquid. Okay. Next one we have to discuss here, separation of salt from ammonium chloride. Salt and ammonium chloride, how can we separate? We know ammonium chloride, this type of solids this type of solid will sublimize. Here ammonium chloride, iodine, camphor, this type of solid will sublimize. Sublimize means this type of solid directly change to gas without change to liquid state. They will directly change to gaseous state. Solid directly change to gaseous state. This process is called sublimation. So we have a mixture which contains salt and ammonium chloride. Okay, so you have here one picture. Here salt and ammonium chloride. We have to take salt and ammonium chloride in a china dish and invert a funnel as shown in the figure. Invert a funnel on the china dish. Okay, then put some cotton plug on the tail of the filter funnel. Then heat china dish. We know ammonium chloride, it will sublimize, it will change to vapor very easily, and this vapor will condense on the surface of funnel, but salt will remain on the china dish. By this way we can separate the mixture of salt and ammonium chloride. So here I will say once again, we have to take the mixture in china dish. Okay, invert a funnel as shown in the figure. Invert the funnel. On the tail of the funnel we have to put some cotton plug. Heat the mixture. Okay, after that ammonium chloride will sublimize, and after sublimation we know ammonium chloride will change to vapor. This vapor, ammonium chloride vapor, will condense on the surface of filter funnel. It cannot escape outside because the tail of the filter funnel we covered with cotton plug. So this vapor do not escape outside. It will condense back on the surface of filter funnel. But salt will stay on china dish. Okay, this method can be used to separate salt and ammonium chloride. Also see here, how can you separate salt and iodine? How can you separate salt and camphor? Same process we can use. Okay, next one we have to discuss here, chromatography. Next method of separation, chromatography. Chromatography is a method of separation to separate colored mixture. Colored components or colored mixtures can be separated with help of chromatography. Chrome means color. Okay, so chromatography used to separate colored mixture. For chromatography we will use a special type of paper called chromatographic paper. Okay, also we can do this experiment with the help of normal filter paper. So take out a filter paper. This is the size of filter paper we will use to chromatography. Okay, around 12 centimeter length and 3 centimeter width. Take this type of chromatographic paper, and lower end Lower end of this paper we have to draw a line with the pencil. Okay, pencil. Do not use ink pen, because this chromatographic paper we have to insert into water. Okay, when you draw with ink pen, and it will give you wrong answer, because we have to separate colored substance. Okay, so ink, it contains colored pigments, so there is a chance to get wrong answer. That's why we have to draw this baseline with pencil. Okay. After that we have to put the sample, the mixture, the sample mixture. Okay, we have to put 1 drop of sample mixture on the baseline as shown in this figure. Okay, after that we have to insert this chromatographic paper into a beaker which containing water. Okay, we have to make sure that this baseline should be above the surface of water. Do not insert baseline and sample drop into the water. It should be above the surface of water. Okay, and you can see here in this video, you can see here, I will show you one video after this. You can see here chromatographic experiment. So here in this video you see here this colored components will move upward, it moving upward, because this is filter paper. This water will rise upward through the filter paper, and this water helps to separate different components of colors from the sample which contain different components of color, and it will separate into different spots or different pigments, okay, with the help of this water. This process is called chromatography. Look here at the end, we have to say this is the result of chromatography. We will say chromatogram. This is the result of chromatographic paper. Imagine our sample which contain 2 pigment. So we will get here 2 spot. Imagine our mixture which contain 4 pigments, 4 different colored pigments. So there will be 4 spot. Okay. Here this is our sample mixture. It contains 4 pigments. So it form 1, 2, 3, 4 spot, and this result is called chromatogram. Okay, so these are the things we have to discuss about chromatographic experiment. chromatographic experiment. This experiment used to separate colored components, and here we have to use a special type of paper called chromatographic paper. And here the size of chromatographic paper around 12 centimeter length and 3 centimeter width. And the lower end of this chromatographic paper we have to draw a line with the pencil called baseline, and put one drop of mixture on the baseline and insert this chromatographic paper into a beaker which containing water. Okay, and this water will move upward through the chromatographic paper, and this water helps to separate different components of mixture. Okay, and this process is called chromatographic experiment. This chromatographic experiment, applications of chromatographic experiment. Chromatographic experiment helps to test purity of food and drugs. We can check purity of food and drugs with help of chromatography. Also here we discuss different colors of colored components from dyes. Dyes mean it contains different colored components. We can separate different colored components of dyes with help of chromatography. Okay, pigments from natural colors which contain different pigments. For example, chlorophyll, it contains natural pigment, green pigment. We can separate this pigment with help of chromatography. Okay, so these are the different applications of chromatography. Thank you for watching this channel. Please subscribe and press the bell icon for more videos. Thank you.